We are delighted to bring you this seminar presented by Labroots. Welcome everyone, and thank you for joining us for today's keynote presentation titled Individualized Health. It is my pleasure to introduce you to Dr. Michael Snyder, Stanford Ascherman Professor, Chair of Genetics Director of the Center of Genomics and Personalized Medicine. Dr. Snyder received his Ph.D. training at the California Institute of Technology and carried out postdoctoral training at Stanford University. His laboratory study was the first to perform a large-scale functional genomics project in any organism and has developed many technologies in genomics and proteomics. For a complete biography on our speaker, please visit the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them in a Q&A box. Our speaker will respond back to you via email. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Snyder. I will now turn the presentation over to him.
Well, thanks for having me here. Today, I want to tell you about our research that's trying to use big data to improve healthcare. So I'm going to start with a premise that may offend many of you, which is the way we perform healthcare these days is entirely wrong. The next slide shows what I mean by that. Basically, if you look on the left, currently medicine is really focused on treating people when they're ill. As such, it's very reactive. We typically measure very few things when people are healthy. Also, when people are healthy, the frequency with which you measure them is very rare. And nearly every decision about your healthcare is based on population-based measurements. I'll argue that we should really be focused on keeping people healthy, as such be very proactive. We're capable of measuring many, many more things than we currently do. The frequency, I'll argue, should depend on what your disease risk is and your personal trajectories as you're following various biomarkers. And perhaps most importantly, nearly every decision about your health should be based on individual-based measurements, not on population-based measurements. And as such, we should really be focused on precision health. Now, to illustrate this point, with the next slide, this is actually a slide I derived from Stephen Steinhubble. Basically, you're probably taught that your average oral temperature is 98.6. And in fact, if you look at studies, and this is one of a number of them out there, and this is from a fairly large study, 3,000 people. First of all, that number is wrong. It's not 98.6. In fact, 97.5 is a little bit more likely to be the median value. But perhaps more importantly, it turns out if you look at the bottom of the box, that's the 25th quartile. So the 25th quartile is 94.6. And the 75th quartile is 99.1. So there's a range. And so in today's world, if your normal baseline temperature is 94.6, you go to a physician's office today and they measure you at 98.6, they'll tell you everything's fine, you're perfectly healthy, what are you doing here, go home. But in fact, you're up 4 degrees over your baseline. I guarantee you're not healthy. You're probably quite ill. And there's no way they would know that because they don't know your individual baseline. So this is why I would argue it's really, really important to know your healthy baseline, and then you'll be able to better detect disease in you rather than from using population-based measurements. So that's a theme I'm going to hammer many times throughout this talk. Now, I'm sure you know your health is dependent on many different things. We know intuitively it's dependent on your genome and all these other items that are listed on this slide as well. So in the bottom left, your environmental exposures, stress, food, exercise, these all will impact your health. And we're now in an era where it's possible to quantify these things so we can understand in quantitative terms what sorts of things might impact your health, especially in light of your particular genetic composition. So that is to say, based on your genome sequence, you can see which variants might expose you to certain kinds of predispositions that will also become accentuated with environmental or lifestyle factors. So we can now measure, as I say, most of these, although it's somewhat clunky, but nonetheless, we can measure their biochemical effects by actually looking at our blood and urine and looking at the various analytes. And so as many of you may know, a number of years, it's actually over 10 years ago now, we set up something we call personal omics profiling. And the key elements of this are we do big data measurements on a group of people. It's 109 people, who I'll tell you about in a minute. We do deep omics measurements, and we follow them longitudinally. Those are the key aspects. So in its most deluxe form, we'll follow 14 different omes, the genome, but then all these other omes as well. Many of them, like the epigenome, that's DNA methylation, and transcriptome, those will be measured out of your immune cells, called peripheral blood monocyte cells. And then many of the other things listed there, like the proteome and cytokines and metabolites and lipids, those are all measured out of your plasma. In some cases, like metabolites and lipids, we measure those out of your urine as well. We also measure the microbiome. So as I say, in the most deluxe form, we'll measure 14 different ohms on people, but for the most part, we're measuring about 7 or 8 ohms. In addition to that, we have deep clinical testing. We run questionnaires as people get sampled. On the left side, we actually have smartwatches on people we put about 7 years ago. And we also run a number of advanced tests like stress echoes, glucose control measurements, because we're very interested in type 2 diabetes, as you'll see a little bit later, and how people become type 2 diabetic. And so we have a lot of measurements there. And in fact, this cohort, it actually turns out it's a typical US cohort. We thought we were enriching it for type 2 diabetics in the sense that 9% of the folks who entered are type 2 diabetic, and a number of others, roughly about 35 to 40%, are pre-diabetic, as you'll see in a minute. And the key element out of this is that we're actually sampling this cohort in general is pretty healthy, aside from some of them having elevated glucose and sugar. And so we're following them longitudinally. That's a very, very key aspect. We sample them quarterly when they're healthy. And then when an adverse event comes along, like a viral infection or maybe a colonoscopy, we'll typically take 5 to 7 additional samplings. So we're trying to understand what happens when people are healthy. So what does a healthy profile look like? How does it differ amongst different people? How does it change over time? And equally importantly, what happens when people become ill? Can we see the changes that occur, and how extensive are they? And in particular, how do people react to different adverse conditions like viral infections, things like that? And so, as I say, we've been following this group. Actually, I'm one of the cohort. We've been following me for over 10 years, but most of the cohort we've now been following a little bit over 7 years, some more, some less. And so we've collected quite a bit of data on these folks over time. So why are we doing this? Again, we're trying to understand what it means to be healthy and how people differ from one another. But we also want to know whether these advanced technologies can really be used to better manage people's health. Can we use these to better catch disease early, make risk predictions, things like that? So I'm going to jump right to the punchline with regards to this last question. It turns out that by following people for a little over 3 years, just for the first part of the study, we made 49 major health discoveries. If you count hypertension, the number rises to 67. And they come in all kinds of different areas. So it wasn't like we're focused on cardiovascular, but we saw cardiovascular, hem-oncology, metabolic discoveries as well. And so 49 out of 109, that's almost half the folks, had something very, very important they learned about their health. And if we look just down at the bottom part in the hem-onc area, we caught someone with early lymphoma. I'll show you examples of this in a minute. 2 people with pre-cancers, one's called MGUS, another's smoldering myeloma. Some folks with thalassemias. In the upper right part, we basically found 2 people with serious heart issues, one from genomics, one from actually wearable devices, plaques in the aorta, things like this. Metabolic disorders were picked up as well, so on and so forth. And the key element out of all this is that no one of these technologies found all these. It really took the combination of different technologies to be able to reveal these health discoveries. And so we think they all add value. And ultimately, we'd like to see some form of this, I'll come back to this a little bit later, incorporated into healthcare. Now, of these different technologies, as I say, they all had value, but one that started off with a lot of value was, in fact, the genome sequence. Just from the first 70 people, we had 12 that learned something very, very important for their health. And as an example, one of the 12 had a mutation in a gene called BRCA1. So you probably know that BRCA genes predispose people to breast and, for women, ovarian cancer. And so one of the individuals did in fact have this, and they will get increased screening as a consequence of this. 3 people learned something very, very important for their health. One person, if you look down the list, you'll see that RBM20, one person had a mutation in a cardiomyopathy gene. And once you know it, it turns out they, actually a pretty young person, they had a heart defect, which was picked up in a subsequent stress echo, and they're on medication now as a consequence. You look at the very top line, 2 people where it says 2X, that means 2 different people, each had mutations in that gene called SDHB. That mutation predisposes people to certain kinds of cancers and tumors. And once you know, one of those individuals does a whole body MRI, turns out had early thyroid cancer, which was caught, and that part was removed, and they actually were able to maintain most of their thyroid, so they don't need thyroid replacement therapy. And finally, as I mentioned, there were actually 9 diabetics in the study. They were all thought to be type 2, but one individual, if you look at the case last from the bottom, HNF1A, that actually person turns out that they were thought to be type 2, but it turns out they have a mutation in a MODY gene. And that's a very different kind of diabetes than type 2 diabetes. So in fact, this is a MODY case, and they've been on the wrong medication for years. We also found some with a very, very important variant that affected a drug they were taking. So the bottom line out of this was that we had 12 and 13, if you count the pharmacogenetic finding, 13 people who learned something very, very important from their genome. And in several cases, it really had critical impact on their health and the drugs they're currently taking. So to show you a few other examples, this is a case at the top of a person with early lymphoma that was actually picked up with imaging. They had a large spleen. If you look at top right, it turns out their spleen was much larger than normal. And they also had several other biochemical markers that were off. In the bottom part, there were 2 people I mentioned who had precancers. These are picked up with biochemical assays. One individual, also a fairly young person, turns out they'd given 2 samples, and those 2 samples had very, very high levels of a protein called IgM. It's an antibody, and it was just way higher than the other 1,000 samples that are at the bottom of the plot there. So they were just way off relative to everyone else, and then follow-up studies revealed that, sure enough, they actually had a precancer. And this is a big deal because they'll get screened more frequently to see if they'll convert to aggressive cancer, and that could get caught at a much earlier stage with this information. We also had 6 people who had plaques in their artery. That's shown here. I'm actually one of the 6. These are relatively modest, but it's kind of useful to have caught this because the follow-up is that they should have their statins increased. That was true for me. My plaque is now dissolving. So again, this was very useful information to find. As I mentioned before, there is a big emphasis on trying to understand type 2 diabetes. And we did have 9 people who thought they were diabetic when they entered the study. That's on the left bar in the blue, so not 9 or 8.4%. But it turns out 2 people were diabetic who didn't know it. They found out when they entered the study. And then quite a few people became diabetic during the study. In fact, 9 additional folks were clinically classified as type 2 diabetic as the study progressed, and others had diabetic range values. And what we were very interested here was understanding how do these folks become type 2 diabetic? That's never really been explored before. People just kind of get classified, but how do they actually get there? And it turns out that, sorry, this it's showing on mine. Maybe we could click on this here, Tyler. Something's funny. We're missing a panel. It's an animation.
This is in slide 10.
Yeah, sorry about that. You'll have to cut actually this part. So do you see that? Yeah. You can animate on it, but I cannot apparently. You don't see that. So the right side shows in my mini panel, but I didn't catch that before, but doesn't show in the big panel. Yeah, there it is. Okay, so can we start here?
Yeah.
Or we
do
can just...
Do you want to start from the top of 10? Or...
Yeah, why don't I start from the top of 10?
Okay.
Okay, you ready?
And do you want to keep the slide like it is right now?
I think so. Then we don't have to worry about clicking through it, if that's okay with you.
Okay, yeah, no problem. Let me go ahead and count you down then from 5, and then you can start from the top of 10. Alrighty, starting in 5, 4, 3, 2.
We also had a number of people who were diabetic, as I mentioned before. 9 were thought to be diabetic when they ended the study. And it turns out that when we looked at this further, when they first enrolled, we discovered 2 others were diabetic who didn't know it. So in fact, 11 were diabetic at study entry. And we're very interested in how do people become type 2 diabetic. So if you look at the left side, the blue part, it turns out that 9 others, in addition to the first 9 others became clinically diabetic during the course of the study, and others had diabetic range values. And as I say, we're very interested in how do people become type 2 diabetic. That had never been explored before. And by the way, a lot of people were pre-diabetic and I had no idea. That's not surprising. 9 out of 10 pre-diabetics have no idea. I guarantee a lot of you watching this video are pre-diabetic, spiking your glucose away and have no idea. Anyway, and then others became pre-diabetic during the study. But we're very interested in how to become type 2 diabetic. And it turns out, as I say, there were 9 who became clinically classified. Of those 9, 2 got there through weight gain. That's the classical way. People gain weight, their microbiome diversity drops, and their glucose goes up. And that's how 2 individuals became diabetic. But 7 others did not gain weight, yet still became diabetic. In 5 of the cases, they just gradually became diabetic. And if you look on the right side, the bottom 2 plots, the orange line is in fact fasting glucose, and the gray line is actually hemoglobin A1C. And then there are these numbered blue dots. Those are actually oral glucose tolerance tests. And in the bottom right plot, that individual was first classified by oral glucose tolerance test. They had very high values. They're spiking their glucose. The one in the middle actually was first classified by fasting glucose when they went out of range. And they steadily became, that's true for both of those folks. But there were 2 additional people who didn't gradually become diabetic. In fact, they spiked their way there. I'm one of the 2. In fact, the top left is me. So initially, that's about 8 years of data sitting there, maybe a little bit more. Initially, I first became, if you look at the left side, type 2 diabetic after a nasty viral infection, respiratory syncytial virus. And I happened to go into the physician because of my genome, I predicted it actually. And I went to a physician to get it measured. I was actually getting something that would measure insulin resistance. What we discovered was my glucose was abnormally high, and actually a follow-up, it was very clear. It spiked into diabetic range. That little bump on the left, it looks small, but it's over a year of data. In fact, I had to change my lifestyle. I cut out all my sugars, and I had been biking, but doubled my biking, started running, and I actually got it down to normal range. In fact, it was so boringly low that I stopped looking at it. And then I really didn't look at it all. Somebody else looked at it. If you look at about in the middle of the plot, somebody else looked at it and said, Mike, your hemoglobin A1C is 7.1. You're back to type 2 diabetic again. So I had no idea. So they sampled me a few times. I'd stopped running, and you can guess when. See where it says viral infection on that second one? I'd actually stopped running then. So it's not clear whether the viral infection or the lack of running caused that spike in glucose and hemoglobin A1C. But anyway, it spiked up again. And so after it got up to that 7 range, I started running. I could get it down, never all the way down the baseline. And then it gradually just kept increasing. So what I did as a consequence was I had been running quite a bit. That's how I got it from 7.1 down to about 5.7. And it just gradually kept creeping up, even over the years. And was probably running about as much as I could 4 days a week, roughly 4 to 5 miles a run. And nonetheless, it kept creeping up. And so about 3 years ago, I switched from running to actually weightlifting because I had heard muscle mass is better for glucose homeostasis. And I thought that would help, but actually didn't. It kept creeping up at the same slope. So in fact, that didn't work. And then finally I said, well, it's time to bite the bullet. I took metformin, which is what you normally would take for type 2 diabetes. And I did, in fact, take that. And wouldn't you know it, I'm a non-responder. So in fact, my glucose kept going up all the way to 7.5. And I'm like, oh no, this is trouble. So what I did, I wasn't sure what to do next, and actually we had other data, and that's what really revealed what was going on. It turns out that we can measure, this is a complicated graph, but we can measure my glucose disposition rate as we did for everyone else in the study. And what we discovered is that I'm actually insulin sensitive, which is a little unusual for a diabetic. I'm also making insulin just fine. So what's wrong with me? My cells respond to insulin, I make insulin. Well, it turns out I don't release it from the pancreas properly. That does happen, it's not that common. And so my glucose disposition, I'm one of these people at the very, very bottom where I actually am very, very slow about getting rid of my glucose out of my blood because I'm not releasing my insulin from the pancreas. And it turns out there's a drug for that. It's a prandan derivative called rapinolide. And it turns out with this information, I took that and it worked like a charm. It drove from 7.5 down to 6.3 within a few months. So that's a really good sign. And all this worked out because I actually could figure out what was wrong with me. And so this is what we call precision diabetes. The goal was to know exactly what's wrong with people so you could treat people with the right drug and get the right outcome. This drug I'm taking, it's not a second or third line drug. It's much further down the list. And so it's probably very helpful in my case because I know exactly what's wrong with me, and so I can drill in on that quickly. So we would argue that this kind of information should be done for everyone, quite frankly. This is not a hard test to do to figure out how people are diabetic, and maybe we can put people on the right drugs better. Now, one of the most important things we've learned so far from this study is actually summarized on this slide. What we've discovered is that, in fact, people have very strong personal characteristics. So no matter which ohm we use, whether it's your metabolome, your proteome, your microbiome, cytokines, clinical labs, you actually have very, very strong molecular characteristics, and they don't change that much over time. So what this slide is showing, just for simplicity, it's showing 12 people with at least 10 healthy visits. And every dot is a different visit. Every color is a different person. I'm the light blue person. It's about 6 years of data for me. And what you'll notice is that people cluster according to their own profiles. They're generally, again, the blue dots are together, the red dots for that person are together, the brown dots, that's a different person. They're all together, and they don't change that much over time, even with 6 years of data. Now, the transcriptome, I know, looks a little more jumbled, but if I were to show you that 3 dimensions, you'd get the same result. So the point out of this is that we all have very, very strong molecular profiles, and they actually don't change that much over time. Moreover, it turns out if you undergo some sort of serious perturbation, say a viral infection, we've shown by exercise, we've now done this by a lot of different ways. This one here is actually people who did a weight gain, weight loss. If you undergo one of these, we call them perturbations, you will shift your profile. No question, your microbiome will change, your metabolome, but you will still look more like you than anyone else. So these profiles are very, very robust, and that's shown here. This is just looking at microbiome after weight gain, weight loss. There are 9 people here. Each color is a different person. We've, again, clustered them by similarities, in this case, in microbiome, and they cluster by person. So you'll notice at the bottom, the green person clusters together. Above it's the brown person. All these folks did a weight gain, weight loss. They actually gained on average 6.5 pounds. But again, they didn't shift they shifted their microbiome a little bit, but they still look more like them than anyone else. So why is this a big deal? Well, we think this is why you need these personal longitudinal measurements. If you're trying to just compare your disease status relative to everyone else, that signal is tough to find. And that's because your personal profiles, again, are so strong that you can't really, your disease state and your healthy state are closer together than your disease state for you closer to everyone else's disease state, for the most part. There are some exceptions. So the point out of all this is that this is why the GWAS guys need tens of thousands of cases and tens of thousands of control, because that signal is very, very small when you're trying to look at disease versus not disease. It's much, much smaller than if you look in the same person where you see their healthy baseline and find their disease state. There, you can't miss that signal. You can't miss that delta. And if you think about it, that's what precision medicine is all about, individualized medicine and health. We want to understand at a personal level when people shift, and you're going to have a hard time finding that from these population-based measurements. So this is why these longitudinal profiles, get your healthy baseline profile now while you're healthy, so you can follow then your disease when it does shift. Now, as I say, these profiles are generally quite stable, and that is true. But they will gradually change. There's a few molecules that will change over time. And because we've been sampling people so much, we can actually see what markers are changing on the same people over time. And it turns out that at a cross-sectional level, meaning look at old folks and young folks, we will see plenty of markers that are different between old people and young people. And we find a lot of the same markers people have described already, but we find a whole lot more. But most importantly, in the same person, we can see exactly how they're changing, i.e. aging over time. And this is just two examples. I'm the guy on the left, there's someone else on the right. So in person one, which is me, it turns out I'm a pretty typical ager. It turns out that my coagulation system, the molecules shift up over time. That's known already. Some of my metabolic markers are changing, and liver and kidney markers actually also gradually changing over time. But the cool thing is you can measure this in an actionable time frame, meaning you can actually do it within 2 years, see how people are aging. The person on the right, it turns out their major pathways that are aging are actually cardiac pathways. So their heart pathway is actually shifting over time in a very statistically significant fashion. Most of these markers for heart function are changing. So we had enough people, we had 43 people that we could actually see how they were aging on time. And we said, well, what are some of the major categories by which people age? And so using informatics, we could group people into 4 what we call ageotypes. So ageotype is how you're aging. Now, there are more than 4. It's just that there are 4 major classes that got teased out when you only had 43 people. And some people, the top part, they're kidney agers, liver agers, the second line. Each column, by the way, is a different person. Each row is their ageotype for that function. So kidney, liver, metabolic, immune. And so some people, if you go all the way to the right, some people are aging in all 4 categories. If you look at the rightmost box that's got a red box around it, that person is aging in 3 categories: liver, metabolic, immune function, but not so much in kidney. And then if you go all the way to the left box, is the one all the way to the left, that person is, the left box is a kidney and metabolic ager, and the one next to that, that person's a kidney ager, and so on and so forth. So the point is, we can see exactly how people are aging, and we think some of this information is clinically actionable because some of the markers in this are actually clinically actionable markers. And in fact, there are a total of 6 clinically actionable markers, although we think the biochemical pathways are a better description of what's going on. But nonetheless, for these clinically relevant markers that are shown here, we can follow at a personal level how people are aging. And so the left one is this hemoglobin A1C, a measure of diabetes and glucose function. And if you look at the rightmost bar there, so within each box there's a bar with a red dot over it. That's actually what the group as a whole is doing. They're actually going up in their hemoglobin A1C, their BUN, which is neutrophils, creatinine is what the third box is, should be creatinine, not creatine. They're actually, everybody's going up. And this is known already, this happens over time. But if you look at individuals, if you look at the bars, like just look at hemoglobin A1C, what we've discovered is that some people are going up, as you would expect, but there's actually some people who are going down. That is to say, they've actually decreased this metabolic marker, and that's a good thing in the case of hemoglobin A1C. And in some cases, we have a molecular explanation for this. They're actually started exercising, or they lost weight, or what have you. So for the case of hemoglobin A1C, we could actually explain it. And this is kind of cool, because what it means is if we define your ageotype, you can take action and you can change that, which is pretty good, we think. Now, over on the third box is creatinine. It turns out all those dark bars, those people are statistically significantly dropping in their creatinine, which is a measure of kidney function. So they're improving their kidney function, which is pretty cool. It's not clear what's going on there, but there are 10 folks that have statistically significantly improved their kidney function, and 8 of the 10 are actually on statins. So we think that this is there's something about statins that's probably improving their creatinine function. It might be related to the muscle mass and things like that, but we don't notice anything obvious there. Perhaps they're also improving their kidney function. So the point out of all this is that for the first time, we can see at a personal level how people are aging, and then actually it's potentially actionable. And if you think about it, there's a ton of supplements out there that are on the market, and you have no idea what's good and what's snake oil, and you would have no way with current technology of figuring out how you're aging. The telomeres, they just change too slow, they're too noisy. It's a terrible assay for aging. Whereas with these personal aging markers, you can actually see for the first time how they're aging. You can actually act on it and see if you're reversing any of those. So this is a lot of the in-depth omics profile that we've been doing. It's really turned out to be quite powerful. Now, one issue that comes up a lot is, how do you scale this thing? And so here's where I'm totally conflicted, but we formed a company called QBio that's actually basically doing a medical version of this of IPOP, as we like to call it. So it's spun out, and it's not collecting as much data, but it's more medically actionable data. And so it does deep profiling people longitudinally, and it also does whole body MRI, which the research project we're doing, the IPOP one, does not do. And even right off the bat, we've done, just from the first 100 or so patients, we've done many more now, participants, I should say. We found some pretty important things. So, for example, and it often takes multiple measurements. One measurement doesn't always do the trick, but like we caught someone with early stage leukemia, another person with fatty liver and diabetes. We caught someone with prostate and ovarian cancer. And not only that, just like in IPOP, the longitudinal data is very, very valuable, because if you can see how people are changing, you can actually see what's going on. So as one example, we actually caught someone with early pancreatic cancer by following their changes. And that's a big deal. Many of you probably know pancreatic cancer is lethal. So finding this at early stage, we would argue, is a super big deal. So the bottom line is, I know it's so expensive. You have to pay $3,500 for this. I think this is just how it starts, but I'm sure we can get it much, much cheaper. But we think this concept of bringing these advanced technologies to better follow people while they're healthy will be huge for improving healthcare. And so this is an area that we're really trying to push out there. Now, one important aspect of what we're doing is actually the wearables, and it's very relevant in today's pandemic, actually. So many of you may know there's a lot of wearables out there now, and at the time the slide was made, which was about a year ago, there's over 400 of these devices, and they measure all kinds of things. If you look at the right side, they'll measure your activity, meaning your steps, your running, that sort of thing. They measure your heart rate, skin temperature, sleep, blood oxygen, so on and so forth. They measure all kinds of different things. And so the way you use these is somewhat similar to omics profiling, but what's powerful about them is they're measuring things on a continuous basis. And so the way you use these is actually you start getting people's patterns, and then you look for deviations from those patterns. And so looking here, this is actually my profile. This is looking at sleep on the top, heart rate in the middle, skin temperature at the bottom. And you can see I sleep at night, awake during the day, occasional nap. That's why there's a little bump in there, and back to sleep at night. Heart rate is low at night, high during the day, back down at night. Skin temperature, which is different from oral temperature. For most people, it's high at night, comes down during the day, goes back up, and so on and so forth. So you can actually determine people's patterns. And so, as I say, we've been using these as health monitors, following people's patterns, and it turns out it's pretty cool. For me, one of the biggest deviations is actually, it turns out, when I drop my blood oxygen on airline flights. It turns out that's true for everyone. And that's a whole separate study. I can tell you about that later. But the point is actually that when you fly on aircraft, your blood oxygen will drop. Now, it turns out that this is also very, very powerful for picking up disease. And I'll show you one example, and this is very, very relevant for today's pandemic. So what we discovered is that we could figure out whether you have respiratory and other kinds of disease using a simple pulse ox and smartwatch. And so, in fact, I figured out my Lyme disease from just that. So the backstory behind this is I was helping my brother put up fences in rural Massachusetts. And then 2 weeks later, I was flying to Norway through Frankfurt. And on that last flight, which is a low altitude plane, if you look at the left, normally on these low altitude planes, these small regional jets, as I like to call them, basically your blood oxygen will drop, but it doesn't drop all that much. It drops about 96 or so, 95, 96. And then when you land, it comes back to normal. Well, in this particular flight, my median blood oxygen was 90. I measure myself all the time, as you probably figured out. And when I landed, it didn't come back to normal. It was 95 or 96. So it was abnormally low. And I also noticed that my heart rate was elevated at the time. And then later learned my skin temperature was off. And if we look at the right side, these are low resolution measurements of that. So the top is heart rate, daily heart rate. The bottom is skin temperature. And this is laid out schematically, but basically I realized that, again, my heart rate was elevated. I later got a low-grade fever. I went to a physician in Norway. I warned him it might be Lyme disease because of the timing. And he actually drew blood and saw my monocytes are up and said, yep, you've got a bacterial, in this case, spirochete infection. He recommended I take penicillin. I said, no, I need doxycycline. And it was tense for a few moments there. But he did give in, gave me the doxycycline, cleared it right up. And then when I got back, it was roughly 10 days later, I basically had my blood drawn. Sure enough, I was Lyme positive. And it's a very well-controlled experiment. I happened to have given blood several days before I left, and I was Lyme negative, so I had seroconverted during that time. So the point out of all this is that I was able to pick up my Lyme disease from a simple smartwatch and pulse ox, which measure your blood oxygen. And so that prompted us to look at all the different days I've been wearing my smartwatch. It's about 2 years of data at the time. Now it's about 7 years of data, maybe 8 years. I guess it's 8 years. We have a ton of data. And the point out of all this is that if you look at the left side here, it turns out during the 2 years I've been wearing the smartwatch, there were 4 times when I got ill. There's the Lyme time. Those are the triangle days, if you will. And there were 2 times when I definitely had a viral infection. That star and the square at the bottom, we now know it's a viral infection. And the one with the circle was a time when I was actually ill. I had high C-reactive protein, or CRP, but I didn't report being ill. So wasn't necessarily strongly symptomatic to the point where I called being ill. But it turns out my CRP was just as high as any of those other times. And the point out of this plot is that if you look at what's there, every single time I was ill with high CRP, I had a higher percentage of outlying skin temperature and heart rate, every single time. And so the point out of this is that skin temperature and heart rate can tell you when you're getting sick, we think, just because you'll have a higher fraction of outlying measurements, and you can do it by other means as well. So that prompted us to write an algorithm that we call change of heart. And we think we can use it to tell when people are getting ill before they realize it, because what happens is your heart rate goes up before you're symptomatic, before you're congested and such. And so on the right side, those are what we call delta plots. They're a change from your resting heart rate. And you can see they go up. You see those little red arrowheads. They basically show where you see this change from resting heart rate. And it's always ahead of when people are symptomatic, and certainly by the time they gave blood. And so these 4 times we saw every single time. And then we had 3 people, one of whom got sick twice. And it turns out the algorithm works great on them. So it doesn't work so well for skin temperature. We think because people wear the watch, some wear it loosely. It works for me. I wear mine tight. It doesn't work for anyone. But the heart rate works for everyone. So we're now actually, as you might imagine, running a clinical trial. Love to have any of you listening join that trial. If you have a smartwatch or wearable, please sign up. You can find it from our website. Look for Innovations Lab, or just write to me and I'll steer you to it. We'll enroll you in our study. And the goal is to really see if we can tell when people are getting sick, and we're specifically gearing it towards COVID-19, but for any illness, we want to be able to see if we can tell when people are getting ill before they realize it. And maybe it'll even pick up some of the asymptomatic folks like I was for one of my infections. So we'll see. Okay. So we think, again, smartwatches are going to be very, very powerful health monitors in the future. We've also done a lot in other areas. I'll tell you a little bit about that. We've done quite a bit with glucose monitors. That's shown here. We've been, because of my high interest in type 2 diabetes and our cohorts, very geared in that direction. So it turns out that we put these things called continuous glucose monitors. You put them on you. They stick on you a little bit like a TB test, but you don't take them off. You leave them on there for roughly 2 weeks. And basically, we put these on normal people, pre-diabetics, and a few diabetics. And what we've discovered is that, people have different patterns. So the top spiker, the middle one's a moderate spiker, the bottom one's a severe spiker. So we call these glucotypes, actually. You'll see in a minute. And so it turns out that there's a lot of people running around who think they're perfectly normal. They're very, very severe spikers. And so we've written algorithms. We take those patterns, we write algorithms, and then we classify people into glucotypes. Again, either low glucotypes, if you look at the bottom side, they either don't spike much, so low glucotypes, moderate spikers in the middle, very, very severe spikers at the top. I'm a severe spiker. So we can classify people into these patterns. So the next slide, it turns out that different people will spike to different foods. This is also shown by Aaron Siegel, where some people, we put people on standardized meals, and as one example, some people spike to peanut butter and bread, others to protein bars, some to both, some to just one of those. Nearly everybody spikes to cornflakes and milk. We think that's like poison, so I actually think it probably should be outlawed. It's probably worse than smoking, but the point is that that's a nasty one. It'll spike your glucose for 80% of people. And this next slide, this is a little complicated, but what was very powerful from our study is that we discovered with these continuous glucose monitors, there are normal people who measure normally, if you look at the right side, who are thought not to be diabetic by either fasting glucose, hemoglobin A1C, or even oral glucose tolerance tests. So it turns out they're very, very severe spikers. They don't have good glucose control. So we think these devices are going to be very, very powerful for being able to help catch people who are in this boat. It's actually a very different kind of assay than any of the other standard assays. And so I think it still pulls out glucose dysregulation. It just does it by a different assay. And we think this is going to be really, really powerful for being able to catch pre-diabetics and help them better manage their glucose control. And these devices, by the way, they're over-the-counter in Europe. They're actually, you have to have a physician order them in the US. But my own guess is they'll become over-the-counter here soon, too. So the last wearable I want to tell you about is a new one we've been working on. It's actually the exposometer. So I mentioned before, your health is due to many things. And I felt we could measure most of these things pretty readily, with the exception of the lower left. In general, we really don't know what we're getting exposed to at a personal level. All of you watching this video right now are getting exposed to all sorts of biologicals and chemicals, and you have absolutely no idea what you're breathing in. So what we did was we started looking at airborne exposures. We took an existing exposometer, which measures particulates, PM2.5, as well as temperature and humidity, and we re-engineered it. This is the latest version. Hope you can see this. But this version actually sucks up air. It's actually twice as much as the original one. So the original one basically brings in about the equivalent of what an average person breathes, about 1/15 of what an average person breathes. And so what we're doing is we've re-engineered this to, under the intake valve, it has a pump. Under the intake valve, we have a filter that captures all the particulates and biologicals. And under that, we have a chemical absorbent that will absorb all the chemicals in the air. And what we do is we actually take these samples, they're personal monitors, people wear them, they take them we take samples longitudinally. And typically for me, what we'll do is sample once during the week, sample continuously during the week, one during the weekend, and then every time I go to a new location, I'll have a different device. So we can actually sample from all around the world wherever I've been traveling. And so what we'll do is we'll collect these samples, we figure out all the biologicals someone's exposed to by doing deep next generation sequencing. So we elute off all the biological material, sequence all the RNA and DNA quite deep, as you'll see in a minute. We actually figure out what chemicals people are exposed to by eluting off the chemicals and running mass spectrometry, and then we analyze the data. And in the first study we did, we're now running this a lot deeper. Shown here, there are 3 people who were profiled for quite some time. I'm P1. I actually been doing this now for almost 8 years, but from the first 2 years of data, we collected data over 2 years. The color codes are by seasons. P2 wore the device for about a year, and P3 for about 3 months. And then we have some very strategic targeted experiments we did as well. I'll tell you about in a minute. And so what we do is we collect these, as I say, from the different individuals. This is P1, P2, and P3. And most of the samples, again, are collected in the Bay Area, as you might imagine, so lower left there. But we can also get samples from all around the world. Again, and because I travel a lot, we have samples from all around the world. And as I say, what I'm going to do is tell you about both the biologicals and chemical exposures people have been exposed to. So as I mentioned, we sequenced quite deeply to figure out the biologicals. And in total, we wound up doing over 70 billion reads. It's really quite a deep data set. About 30 million on the RNA per sample and 40 million on the DNA side. And what we've discovered is that over 2,500 different species are detected amongst the whole group. But it's quite a few even for one person. So the one person wearing the exposometer for 3 months had over 1,000 species. I've had over 2,000 from the first 2 years of data. So you're really exposed to a vast wealth of microbes and other things out there. And we've looked at this in detail. That's shown here. So I'm P1, there's P2, there's P3. And this is just at the kingdom level. Each color code's a different kingdom. So red is bacterial, that brownish color is fungi, green is plants, and blue is animals. And so what we discovered is that the exposome is very dynamic. So it changes a lot from one sample to the next. Down below is clustering by sample type. So the plant samples, plant-dominated samples are the left, bacteria right next to that, and brown are the fungal ones. And so basically, you can see what kinds of exposures you get. And so even for all 3 people, the exposome is very, very dynamic. And it's fairly reproducible, too. So the 3 times as in Monterey are those little arrows at the bottom. Those are virtually always fungal exposures. So fungal exposures are quite common, meaning they're the dominant organism that you breathe. So then we went in and wanted to quantify exactly what is it that you're exposed to, how much comes from location, how much from season, and so on and so forth. If you look at on the left of my samples, you can see there's a trend for the Asian samples to be clustered to the right. This is what's something called a PCA-like plot. But the important point is we can quantify it. So for the items we can measure, the major driver of your exposome for the biologicals is location, but seasonal patterns, that's the one with the environmental circle, that's mostly season, that actually is pretty strong as well. So location and season is very, very strong. In fact, there is some technical noise that contributes to this. So we actually looked at this in more detail on this slide. And basically what we discovered is that we took 4 people, we sampled them over the exact same month, and we looked at what their exposomes look like for biologicals. And so in this case, I'm actually the red person. The red dots are mine. And I travel a lot, so my dots are a bit scattered. My samples are a bit scattered. The person who in light blue lives and works across the bay. Green is pretty close to me in a town called Sunnyvale, a few miles away. And that dark blue is actually a person who lives and works, or purple, lives and works in San Francisco. So the point out of all this is that even though people are in the same general area, we all have very, very different exposures. See the green person and the dark blue / purple and the red, we're all very, very different in our exposures. And not only that, you can quantify where those differences come from. So down below, you'll see those are the organisms that are actually more prominent in the San Francisco sample. So you'll notice one of the top ones is sludge bacteria, is very, very dominant in the San Francisco samples relative to the other ones. If you've ever been to Market Street in San Francisco, you understand this result. It makes a lot of sense. And then other things as well. There are pathogens in the samples. They're very, very low levels. It's unlikely they would cause any issues unless, of course, you maybe were immunocompromised or something like that. So the point out of all this is that there are very, very strong location patterns. Okay. It turns out that there's also strong seasonal patterns that's shown here. So basically, it turns out hundreds of species have seasonal patterns. And so the green ones are the ones that peak in spring, light blue in summer, red in fall, and the more dark blue ones are actually peaking in winter. And some examples are shown on the right. So pine peaks in late April. That was early summer. Late April. So yeah, late spring, early summer. This fungus called turkey tail peaks in late fall, early winter. So comes up late, and so on and so forth. You can find all kinds of things. And then you can correlate these, by the way, with biological effects. And we've just started that. There's a lot more to do here. But one of the interesting correlations for me was that I had mild allergies, and they correlated best with eucalyptus. I assumed they were always pine. In hindsight, it made a lot of sense they'd be eucalyptus, which peaks roughly around the same time as pine. The reason I probably should have figured out before is that actually I'm much more allergic in California than I am when I'm on the East Coast, which has very little eucalyptus. So the bottom line is you can actually make these interesting correlations. So we've also looked at the chemical exposures, and just to give you a quick summary here, chemicals are also heavily influenced by seasons, and so just two examples shown here. The top one tends to peak more in spring, less in summer. The bottom one more in winter, and lower in the spring, and so on. So you can make these. One of the most important things we learned about chemicals, though, is that they're everywhere. So there are plastics everywhere. This compound used to keep insects off called DEET, that turns out to be in all the samples. That surprised me. Quite reasonably abundant in those samples. Pesticides are everywhere, but they're much more in some locations than others. Carcinogens are also everywhere, like diethylene glycol and a few others. So again, but they're in some locations are much, much stronger. We think location is the number one driver of your chemical exposure. There are some interesting patterns we see. This next slide just shows a chemical that correlates with rain. So you may know, you can smell when it's going to rain. That's because of that top compound, geosmin, which it actually comes out of the ground. It's a volatile, so the air pressure drops when it's going to rain. And so what will happen is that this compound will come out of the ground. You can smell it. That's how you know it's going to rain. And everywhere where that bar is up, that was a rainy day. And it turns out pesticides also come up. They must be volatile and coming out of the ground as well, would be my guess, when the air pressure drops when it rains. And other things like plastics go down. They probably got washed into the ground, would be my guess. And so those exposures are less when it rains. We also could correlate biologicals and chemicals, and this is one of the most interesting results. It turns out we discovered there's an anti-correlation between fungal exposures and this nasty chemical called pyridine, which is usually found in paints than other locations. And so if you look at this slide here, the bottom row shows pyridine when and where it's most prevalent, and the upper row shows the fungal species. And you'll notice everywhere where pyridine is low, fungal exposures are higher, and everywhere where pyridine is higher, fungal exposures are lower. So we assume then that pyridine is probably killing fungi. And so this is actually kind of useful to know, because maybe if you're allergic to black mold, you'd actually want to have your house painted with a pyridine-based paint, and you could actually keep off the mold, and that might be very nice for you. Now, you may die of cancer because it's kind of a nasty chemical, but nonetheless, you get the idea. It's a potentially very useful thing to know depending on your health situation. So we can actually take all this information and assemble personal exposure clouds. This is mine. Everybody has their own exposome. So this is mine. A lot of human-centric and mammalian-centric species. So bacteria in your skin show up in these exposomes and stuff. And but there's environmental ones as well. My house has a cat, dog, and used to have a guinea pig, and we picked up all those sequences. inside the exposometer. So you can see, we know who has a dog in our study because those dog sequences pick up. And you can see what plants and insects and other things people are getting exposed to. Now, one of the coolest things we do with all this data is we bring it into a personal health dashboard. So we built this, spent quite a bit of time. We even have an app that you can enter certain things that you're doing, like your health situation, feeling good, feeling bad. But we can automatically pull in all your wearable data as well as upload clinical and other data types and bring this right into your smartphone. So your smartphone, I'm going to argue, will be your most important health device in the future. This thing is going to be just as powerful, possibly more so than your doctor. But your doctor won't go away. They'll still use this. But that's the thing. They can use it to help manage your health better because it can bring in all your information and you can actually follow it, the different data. You can enter different data types like heart rate, microbiome, clinical labs, and see what it is you want to track and quickly see what's going on. And so the point is that actually, just like your car dashboard, it gives you very relevant information right at your fingertips that you can use and you can act upon should you so desire. And then the next slide, this, as I say, isn't just for you. You can actually share it with your physician. And we think some of the data that you get from your wearables is even more accurate than what you'll get in the doctor's office. And ultimately, what I'd like to do is see all these technologies brought into healthcare and be able to better manage people's health. So I'm a believer people will be getting their genome sequenced before they're born. And then together with that more detailed biochemical measurements, so the so-called omic measurements, and sensor information, we can better manage people's health. We can predict disease risk better, catch disease early, and again, hopefully manage things so we can shift from sick care into health care and really try and keep people healthier for a much, much longer time. I happen to have a very, very amazing lab that's shown here. So these folks in my lab are listed to the left. I won't have time to go through all of them, but Wen Yu Shao, Brian Penny, Kevin Contrapa, Tejas, Sophia Rose. I don't see her listed there. They were all very, very powerful in helping getting the study started and going. We have amazing collaborators. George Weinstock helped very fantastic for the microbiome work. Tracy McLaughlin on the clinical side. People who built some of the personal health dashboard are listed to the right. Amir in particular and Raj, I want to give a shout out to them, expose them by the team down below. And it's just been an amazing group of people coming together to try and transform the way we currently do healthcare. So thanks again for having me here. Happy to take any questions online.
Thank you, Dr. Snyder, for that outstanding presentation. For additional informative presentations, view our agenda on our events page. As a final reminder, our speaker will follow up with any questions you have submitted via email. Thank you again for your participation.